Good morning, everyone, and thank you all for attending this presentation today. My name is Sara, and I'm from Faculty of Health and Environmental Science. It's been 4 months that I've started my PhD, so during my presentation, I will mostly talk about my PhD proposal, and then I will go through some of the experiments that I managed to do in the last 2 months. The title of my presentation is Encapsulation of Probiotics for Aquaculture Application, and this research project is being conducted under supervision of Dr. Ali Seyedehsara, my main supervisor, and my co-supervisor, Prof. Andrew Alfaro, and Dr. Naomi Gutierrez from the School of Science. New Zealand is famous for many things, for its Maori culture, for its beautiful natural landscape, but when it comes to the seafood, it's the country of shellfish production that sets it apart from the competition. And PAWA industry is an important part of this shellfish production. But what are the current challenges and problems in this industry that can affect on the New Zealand economy? So I will answer to this question during my presentation, and then I will talk about our proposal about how we're gonna help to overcome this problem. Black-footed abalone, or Hydrolysis iris, is endemic to New Zealand, and it is called PAWA in Maori language. There are 3 different species that occur naturally in New Zealand, but only black-footed abalone is cultured for export market. And PAWA shell has the most colorful shell among all abalones around the world, and because of its natural blue color, it's been used for a long time in Maori artwork, representing the eyes in Maori traditional carving. But recently, some of the researchers started to extract this natural, beautiful blue color, and it's been reported as a relatively stable blue color. So this natural pigment has a huge potential to be used in different industries, such as food, beverage, and cosmetic. So the demand for PAWA shell now is increasing, and its market is developing rapidly.
Sorry, this one's the Chechen mango.
Okay. Yeah. But rather than PAWA shell, PAWA meat also has a large market, and PAWA meat is exported as fresh, canned, and frozen meat with a high price of more than $100 per kg of animal. So PAWA is an important high-value export item in New Zealand aquaculture industry, and there is a strong overseas demand for PAWA, especially from United States, Japan, and China. In terms of economic point of view, PAWA industry is worth more than $60 million in revenue, and it's expected to reach to $1 billion within the next 10 years. At the moment, OceanZ Blue has the largest PAWA facilities in New Zealand, and they produce more than 100 tons of PAWA each year. So the whole PAWA industry is an economically important industry for New Zealand. But what are the current problems in this industry? Abalone are known to have a very slow growth rate. The long cultivation time for abalone that abalone requires to reach to the market size now is a serious bottleneck in this industry. And this long cultivation time, which is around 4 to 5 years, has resulted to serious financial constraint in PAWA industry. So any strategies that can reduce this time frame would confer a huge benefit to the whole industry. So far, different strategies have been used to promote growth rate of abalone, such as using different design of the culture system, and also application of feed supplements into animals' diet, and using probiotics. But among these 3 different strategies, using probiotics offers some advantages over the other 2 methods. By using probiotics, rather than promoting the growth rate of animals, we can also induce the immunity and general health of abalones. So using probiotics could be a great strategy, not only to improve the growth rate of animals, but also to boost up their immunity and prevent spreading disease inside abalone's farm and abalone culture. And also using probiotics can be a great alternative for using chemical additives or veterinary medicines in aquaculture. There are two main studies that they used probiotics to improve growth rate of abalone, and they reported 99 and 20% improvement in growth rate of abalone within 2 months feeding trial using different probiotic species. But what is the what are the current problems related to using probiotics in aquaculture? The main problem is referred back to the conventional delivery method of probiotics to aquatic animals. In conventional delivery method, usually the probiotic is getting mixed with animals' feed, and then the feed is placed into the water. But when we talk about this method, we need to consider the drawbacks that is related to this conventional method. The first concern is that this method is not environmentally friendly. When we mix the probiotics with animals' feed and we place it into the water, we actually release a high concentration of probiotics or unwanted bacteria into the environment. And since the probiotics are species specific, meaning that one bacterial species could act as a probiotic for one aquatic animal, but at the same time it could be pathogenic to others. So we don't want to release a high concentration of unwanted bacteria into the environment. The second concern is about probiotics viability during preparation. If we want to mix the probiotics with animals' feed, we need to consider that most of the probiotics are so sensitive to industrial process, so they cannot withstand the harsh condition during feed preparation. And the last concern is about probiotics viability after being consumed by animal. So many probiotics are so sensitive to the environmental condition during within the animal's gut tract, and for probiotics, it's very important to remain viable when they reach to their target site in animal's body in order to produce the beneficial health effect. Our hypothesis is that by using encapsulation technology, we can actually overcome all those problems. Encapsulation of probiotics could help in protecting the environment from receiving a high concentration of unwanted bacteria. We also can protect probiotics against adverse environmental conditions, and also we can maintain the viability of probiotics by encapsulating them when they reach to their target size in animal body. And also we can have a controlled release of probiotics when they reach to their target site in abalone's body. The objectives of this study are to check biochemical characteristics of probiotics and also to develop a formulation for encapsulating probiotics. And then we will feed those encapsulated probiotics to animals to evaluate the effect of encapsulated probiotics on growth rate and survival of abalones. The methodology of my whole PhD studies will include 3 main stages. The first part is some microbiology work. We will test some biochemical characteristics of bacterial species that we are going to encapsulate. And then we will proceed to second and main stage, which is probiotic encapsulation. We will use a polymer or a combination of different natural polymers to encapsulate those probiotics. And then we will design a delivery system to deliver those encapsulated probiotics to abalone and then investigate the effects on the animal. From this point of my presentation, I will talk about some of the preliminary studies and experiments that I did in the last 2 months. We worked with 3 different bacterial isolates. This bacterial species was isolated by one of the students in our research team from abalone's gut tract. So during the last experiments, we cultured these bacterial isolate and then we tested their ability to hydrolyze starch, alginate, and casein, which are components in abalone's feed. And also we checked their ability to produce acid and then investigate their stability within a pH range from 2 to 8. The result of my study and the experiments show that from 3 different bacterial isolates, actually 2 of them showed the ability to hydrolyze a starch and 1 of them could actually hydrolyze casein. As you can see from the figure 2, the yellow zone around each colony showing indicate the lack of starch molecules inside the plate, meaning that the bacteria could actually hydrolyze the starch molecules inside the starch plate. And on the right side, you can see casein plates, the clear zone around each colony showing that they could hydrolyze casein molecules in the casein plate. And for alginate hydrolysis, we used a spectrophotometry method. We incubated the 3 different bacterial isolates in a media that contained alginate. And we checked their ability to hydrolyze alginate every 24 hours during a 4-day study. From the results, among 3 different bacterial isolates, Vibrio could actually showed a significant ability for alginate hydrolysis. Acid production also is another important factor for probiotics. When a bacterial species could produce acid, they could actually be more helpful in feed digestion. Almost all the 3 bacterial isolates could produce acid, but the amount of acid produced by intro was almost double the amount of acid produced by the other 2 species. And the last experiment was pH stability of 3 different bacterial isolates. The result of this experiment is really important to us for my following experiment, which is encapsulation part. And based on the result, all the 3 bacterial isolates showed a good stability in a pH range between 5 to 8, and none of them could be resistant against very low acidic pH around 2. From the result of my experiment, we can conclude that the 3 bacterial isolates have required biochemical characteristics that can assist in feed digestion and to act as a probiotic for abalones. And for encapsulation part, we may use a combination of 3 bacterial species to assist abalones in feed digestion. And in terms of stability to different pH, the 3 bacterial isolates showed enough stability to pH range of PAWA gut tract, which is between 5 to 8. And for my future study after probiotic experiments, I will proceed to encapsulation part. We will encapsulate probiotics in a combination of natural polymers, and then we will proceed to feeding trial to test the effect of encapsulated probiotics on animals. To summarize my presentation, my PhD proposal aims to develop a novel method to encapsulate probiotics for aquaculture application, and also to develop a probiotic supplemented feed in order to stimulate the growth rate of animals. And this strategy will offer some advantages to the current aquaculture practice by reducing the time frame that abalones need to reach to the market size. And hopefully this will result in high power production to enhance growth rate of animals and by preventing disease inside aquaculture. Okay, thank you so much for listening. These are my references, and I would be grateful to take any question from the floor.
Thank you very much, Sarah.
Yeah, I think it's very interesting topic for industry. And I have a question relating to how do you check the effective of that probiotic, just the growth and survival of the animal, or do you think about checking the digestibility in terms of protein and energy?
Okay. Actually, we may want to check the digestibility of to see if the probiotics could help in feed digestion for animals. But actually, the microbiology work that I've done, it showed that the 3 probiotic and bacterial isolate could actually help in feed digestion by hydrolyzing a starch, alginate, and casein molecules that they are present in animal feed. Yeah.
So in the case you check the digestibility, what method you are going to use?
Actually, up to this point, I think the microbiology work might be enough, but maybe we think of checking the feed digestion by probiotics later.
I see one article they use the metabolomic approach called near infrared spectrometry, and it's very useful to check the digestibility and energy absorption of the animal. So I think it will be, if you combine them together, it will be very good to see a bigger picture about.
Okay. So if you want to use that, I will get help definitely from you.
Yeah, that's a very good
Okay. Yeah. Thank you for suggestion.
Thank you for your question. Anybody else?
I have a question. So how does the probiotics affect the shell's color? Does it affect function to the shell's color and the good marketing?
To the shell's color?
Yeah.
Actually, the objective of my study is not investigating on the pigment of the shell, and mostly we're focusing on the improvement of the growth rate and survival of the animals after feeding them with probiotics.
Thank you very much.
Yeah.
Anything else, Robert?
So your research think about you can make the animal grow faster,
Yeah.
something like that. Do you think it's good for make them grow faster? Maybe they lack of nutrition. I don't know.
Yeah. Actually, this is what we are going to do, because the cultivation time of that abalone required to reach to the market size is very long, like 4 to 5 years, and we are going to decrease that not by adding chemical additives that may affect negatively on the animal, but by adding some bacterial species that they naturally possess inside they naturally have those bacterial species in their gut tract. So I think there will be no negative effect from probiotics on the animal.
So totally no negative effect.
No, I don't think so, because they have already those bacterial species in their gut tract. Yeah.
We just have time for one last short question.
It's a very short question. So it normally takes 4 to 5 years to cultivation time, right?
Yeah.
So how long would you expect that the time would be reduced?
Reading.
Yeah.
So that's the question that we're gonna investigate. So we will use encapsulated probiotics, and after feeding trial, we'll do some study to see how much the possible reduction will be.
So that's the whole study.
Yeah, it's one part of the study.
You have to wait 4 years.
All right. Thank you very much for your question. Thank you very much, Sarah.
You're welcome.